We're just to remind you that we're on the 19th year of the Winter Series on Aging, and it's named after Dr. Edward Lefebvre, who was a general internal medicine physician, practiced in private practice in town, provided care for many individuals, and was affiliated with UTMB and was constantly needling UTMB about its lack of a geriatric program. So he played a large role in the development of the Center on Aging, which is why we, and on his death, his family provided a small endowment to sponsor a lecture. We're very happy for the third lecture of this series to have Dean Hartley, who's the scientific director of the National Alzheimer's Association. Dr. Hartley got his bachelor's and master's in neuroscience at the University of Illinois and went on to get his PhD at Stanford. He then went to Harvard Medical School for postdoctoral training and stayed on the faculty there. He was on the faculty at Harvard for somewhere around 12 years or so. And then he moved to Rush in Chicago, where he was on the faculty at Rush Medical School. And then a couple of years ago, he'd been consulting with the Alzheimer's Association, but a couple of years he went over full-time and became their scientific director. His scientific research, which he probably won't talk about given the title tonight on trials, but his scientific research, he's just done some outstanding basic work on mechanisms of pathogenesis, how the disease Alzheimer's comes about, using mouse models and even, I think, fly models. Mouse models may seem a long way from humans, and Dr. Hartley would agree, but it's been amazing how developing an animal model takes a lot of creativity in order to measure memory impairment and things like that, which can be done in rodents, but it takes a lot of work. So he's been focusing more on clinical research, and we're very happy to have him come and talk to us today about the future of clinical trials in Alzheimer's disease. Welcome.
Well, thank you, and I want to certainly thank the organizers and giving me the opportunity to come here. I've just had a great day talking about science. Yeah, your weather isn't very good, but I think last night it was -6 in Chicago, so this is actually warm. But I agree with you. For some reason, it doesn't feel warm. It's nice and cold. So yes, I joined the association about 2 years ago, and I did basic science for the last 18 years before that. And what I'm going to try to do is give you a perspective of really what the association does and how we're trying to make a difference in moving the needle. In other words, how are we going to eliminate or reduce the number of suffering from Alzheimer's disease? So I'm going to give you a few basics here, just because this kind of sets the stage of what problem we're heading for. I also think it's also an important piece because I'm going to ask you all to be ambassadors for trying to end Alzheimer's disease. These numbers are very important for that reason. When you go out and talk to others, they'll understand these. Sometimes they're so large that we can't even put them into context. But we see right now that there's 5 million people living with Alzheimer's disease. Some 200,000 are under the age of 65. We use 65 as the cutoff point. Those that have late onset, 65 or older, early onset, 65 or younger. So you can see this number is only increasing over time. You can see I have Illinois here. Well, in case you want to know about in Illinois, there's 210,000 people with the disease itself. But the most amazing part is looking at up here, age of 65 or over, 1 in 9 people have Alzheimer's disease. By the time you get to 85, it's 1 in 3. So for that reason, we are living longer, so that's the reason we're seeing the rise in Alzheimer's disease. Right now, it's every 68 seconds someone develops the disease. And by mid-century, we're going to be up to 33 seconds. At least down to 33 seconds when we're developing disease. These numbers are amazing to me, and this is, I think, why we're getting noticed in Washington. You can see that there are for every person with Alzheimer's disease, there's 3 people that are caregivers. So there's 15 million people here taking care of them. 60% of those people are still working. You can see that 17 billion hours, and if you do the calculations, what you come up in terms of unpaid caregiving, $216 billion. Out-of-pocket expenses is 34 million. And I like this last number here because something we don't always appreciate is that caregivers also are under a great deal of stress as well. There's an additional $9 billion that they have to pay for in terms of healthcare. That's because they're trying to take care of their loved ones 24/7, 365 days, and that's difficult. And we actually have things on our website in terms of stress tests to make sure that you can kind of take a look to make sure that you're in check. So this is the direct cost right now. We're at, in 2013, $203 billion. That's in terms of Medicaid and Medicare expenses. By 2050, we're going to be at $1.2 trillion. Our healthcare system can't really take this. So this was put out last year by the RAND Corporation, and it showed that Alzheimer's disease is now the costliest disease in the United States, over heart disease and over cancer. That's because of the number of years the person lives with the disease and the cost at the end of the disease. If you're thinking about this going to $1.2 trillion, the question is how we're going to pay for this. So this is just to remind you about the demographics and why Alzheimer's disease is on the rise. And what you can see as we go here in terms of age, that in 1950 we were kind of a pyramid shape. But as times, we're kind of this column shape now. That means we have a lot more people living to the age of 65. That's the age in which they're now coming into being susceptible to Alzheimer's disease. So for that reason, this and the fact that age is the biggest risk factor for Alzheimer's disease, which we don't quite understand, is going to continue to put this on the rise. So to me, this is one of the most important slides that I can show you of how we're going to change this disease. On the left-hand side here, this is the number of people dying from Alzheimer's disease between 2000 and 2010. What you can see on this end here, these diseases are decreasing. Cancer, in terms of breast cancer, prostate cancer, heart disease, stroke, and HIV. We can see that HIV has gone down 42%. Why is that? Because there was a war on HIV. They put billions of dollars into it and now actually have drugs that treat the disease. Alzheimer's disease during that time went up 68%, and it's going to continue to rise. Because it's the sixth leading cause of death. We have no treatments, no cures for the disease. But on the other side here is one of the major issues. If we look at what the federal government has put into spending for research, you can see it's between $4 and $6 billion a year. Way over on the left here is Alzheimer's disease, about $500 million. So you can see, in fact, that the amount of money that's being put in this, even though it's a causeless disease, is really pretty small. So we need to change that. That needs to go up to these levels. We don't want to take away from those diseases because they're just as important. But somehow we have to find the dollars to move the needle. So I like using this slide here. I don't know if any of you saw this movie. It's called The Perfect Storm. I think George Clooney actually was in it. And this was off the coast of Massachusetts. And actually it was the first year that I moved to Massachusetts, and I'm actually at work. I'm at the Children's Hospital in Boston. And my wife calls me and she says, there's this raging storm outside. You have to come home. I'm in the middle of this building. And I said, I can't leave. I have research to do. And I don't care about the storm. So it's about a half an hour later, my wife calls back and she says, you know what? Your bike just blew away. Well, she actually got my notice at that point and realized that it was this storm. It was a no-name hurricane that actually came up late in the season. And it was because there were two different fronts. One had come from Canada, one had come up the Gulf. And where they collided was off the coast of Massachusetts. And that produced this large storm here. And I like to use it for the analogy of where we are thinking about this large tsunami in terms of healthcare costs and the disease perspective, is that we've been having decrease in funding in terms of NIH dollars. That's a problem. And I mentioned to you that Alzheimer's is on the rise. That's, to me, a perfect storm for the problem that we're going to have. And it is here now. The number of people. We have 10,000 baby boomers coming over the age of 65 a day. And they're coming into this pipeline. So it is a major health crisis that's here, and it's going to only get greater. So, what are we trying to do about this? Well, in 2011, President Obama signed into law the NAPA, National Alzheimer's Project Act. It's something that the Alzheimer's Association is very proud of, because we helped craft the legislation for this bill. The interesting part was, it was unanimously voted in. And you know why it was voted in? There were no dollars associated with it. So all the legislators could easily say, I understand the problem with Alzheimer's disease, but you know what? I don't have to pay for it. Well, every year we have a national plan. The national plan is to look at where have we been and where are we going. Our CEO, Harry John, sits on the advisory panel, and they work very hard to make sure that the dollars are going to come forward to pay for these. We need to increase trial recruitment, expand scale and scope of the research, accelerate drug development. Those are all things that are in the national plan. So one of the things the association has done, we started actually in 1979, first chartered in 1980, was to provide care and support. That's always what the Alzheimer's Association can do. But increasing concern and awareness is a big part of making sure the movement, and those dollars are going to come into the coffers to make sure we make advancements in terms of research. We have a public policy group that works in Washington, and I have to remind that even though we are a granting organization, an important part is making sure that Congress appropriates the right dollars to the National Institute of Health, because they do a lot of the heavy lifting in terms of research, and we need that. So our policy groups work very hard in Washington, and in fact, I actually have many times gone to Capitol Hill and talked to legislators, bringing some of those very same numbers to them so that when they get in committee meetings, they actually can have some ammunition to say, that's what we need to know. Those are the dollars. Those are the things that we have to change. So I'm going to talk more about advancing research, since that was the reason for being here. But we are a global leader in Alzheimer's research, and we do that at many different levels. We have roundtables where we have 27 different large pharmaceutical companies coming together and talking about, in a non-competitive environment, what are the problems for Alzheimer's? What do we have to do? What type of reimbursements for our drugs? How do we get looked at? What type of testing? We have our international conference called AAIC, Alzheimer's Association International Conference. It's usually over 5,000 researchers that come together to talk about issues relating to Alzheimer's disease and dementia. We have professional societies. We have our TrialMatch, which we'll talk a little bit later, is a free service to find people who want to volunteer for clinical trials. A very important part of getting the answer to this problem is clinical trials and having volunteers. We have our journal, Alzheimer's and Dementia, and we have a lot of work groups in terms of looking at what is the latest science and what do we have to do to make sure it's appropriately used. So we are the largest private funders of research. We have done $350 million since 1982, 2,200 different scientists. Currently, there are some 350 active grants in 21 countries. That's because we know this disease knows no borders, and we're looking for the best answers. And that's why we're doing it. We have an international grants program that is peer-reviewed at 2 different levels, and then we have a international medical and scientific advisory council that helps adjudicate differences in the grants. But it's a peer-reviewed, very similar to NIH. We have different programs in terms of social behavior, cognitive, functional, and basic science, and clinical. And one of the things that we're proud of is we have a new investigator. The majority of our grants actually go to new investigators. We feel they're an important part of the pipeline. One, they're very vulnerable to the funding side. But two, they bring a lot of new ideas into the science. So we want to make sure that's into the fold. So we have a program in which investigators can apply to specifically that are less than 10 years after their PhD or MD degree. And they also have a cycle that goes through faster to make sure that if they didn't get through the first cycle, they can take those comments and get to the next cycle. So what causes Alzheimer's disease? The answer is, we don't know. But there are some hints. I've talked about age. Age is very important. You can see the likelihood doubles every 5 years past the age of 65. And you can see at the age of 85, reaches a risk of nearly 30%. We know about family history. If you have somebody who's a first-degree relative, you're going to have a much higher risk for Alzheimer's disease. And that's because your genes that you carry are similar to someone that you're related to. You may be carrying the same risk genes. We know that head-heart connection. There's a concern about increasing risk by the fact of stroke, diabetes, hypertension. One in 3 people over the age of 65 have hypertension. So for that reason, we believe a healthy heart is a healthy brain. What we don't know is whether those conditions actually lead to Alzheimer's or whether they help contribute to the dementia in the brain itself. In other words, 2 diseases add up to a greater amount than the individual one. So this is something that needs more investigation, more research dollars. And you've probably heard a lot more recently about head injury. The fact that we've heard about NFL, we've heard about concussions. This is signature injuries that are coming home from the Middle East. They've given them the right apparel to make sure that they don't die, but they come home either missing limbs or having concussions. And being put back into any condition where you're going to have additional head trauma is not a good thing. And this is one of the things that I guess they've been worried about in the NFL because there is this attitude that I have to get back in the game. So for that reason, we're building stronger links to think about this idea. The question is whether the pathology is exactly the same. There are some things that look like they may be different, but this needs to be investigated as well. So what is Alzheimer's disease? It's actually a slow, progressive neurodegeneration, meaning it starts almost imperceptible, and at some point, when you get into a clinical setting, these people can actually use their clinical acumen to say, indeed, we can see those changes happening. This looks like Alzheimer's disease. For that reason, I think it's interesting to know that the brain is going through many different changes during that time. It could be going through biochemical changes, go through structural changes. You can actually see disconnect between various synapses, the way the brain's cells communicate with each other. And for that reason, what you see on the left-hand side is a severe Alzheimer's case. It looks shrunken, and that's because there's cell loss, and there's disconnections between cells, and the processes are being lost. So for that reason, this also underlies the progression of somebody with the disease, first being manifested often in memory problems, then going through many behavioral problems based on the fact that many other systems are being affected, and it moves throughout the brain. And I think sometimes this helps people, especially caregivers, because they don't understand that sometimes when people are getting angry at them or changing in their behavior, there are changes going on in the brain that are responsible for those. So one of the common terms that's used is interchangeably between Alzheimer's disease and dementia, is somewhat correct. But dementia really is kind of the overarching umbrella for the disease or its symptoms. All these underneath here are the specific diseases that have similar symptoms in terms of problems with thinking and social abilities, and dementia really is interfering with daily living activities. So for that reason, Alzheimer's disease is the majority of the dementia that we see, but there is frontotemporal dementia, Parkinson's disease, Lewy body disease. And I think one of the interesting parts is that as we've seen, looking at the pathology of these brains over time, that we realize that maybe there isn't always a pure Alzheimer's case. You can see, we'll talk about a little bit of biology, but there are probably other things going on with this aging process. We've now seen that there are microstrokes, microhemorrhages, that may add to that same pathology. So for that reason, I like to make that distinction because even on Capitol Hill, people are always using dementia thinking they're talking about Alzheimer's disease, and to be perfectly clear, it may not be. So for that reason, we'd like to make that point. One of the things that is really unique about this position is trying to think about those additional pathologies and how can we actually work together. Just last week, the Alzheimer's Association, in conjunction with Michael J. Fox, announced a new program in which they're going to be looking at the data sets that are both involved with Alzheimer's disease as well as Parkinson's disease to see where there's those intersections between the two. And this is an opportunity for us to learn about these additional pathologies and may be very important for our clinical trials, which we'll talk about a little bit later. So, challenges began about 100 years ago, and that's when Dr. Alzheimer's first described in a goose eater, which is this woman here. She was 51 years of age. She was already institutionalized, so you can see that she was looking like a young onset individual. And through the recent, going back through the archives of Dr. Alzheimer's, they actually found tissue from her and realized that she actually had one of the mutations that cause early onset Alzheimer's disease. But upon her demise, this is what he saw. The two hallmarks of Alzheimer's disease in terms of its brain pathology. You can see here plaques, which happen to be made of a particular protein called beta amyloid, and these tangles, which are made of tau. These are normal proteins that you have in your brain. These have normal functions. The question is, what happens to them to be changing? There's abnormal folding of these proteins, and they seem to take on different functions that may actually make them toxic. So for that reason, it's the question of why these start to form clumps. This is actually within cells, and these are outside cells. And this is just a diagram to tell you really how did we get to some of this point. And they were able to grab these and actually isolate them. And if you take and look at them at a very high-powered electron microscope, you can see these type of fibrils. They were then able to take those fibrils and then pull those apart and see what's in it. And this little cartoon here is what we call a transmembrane protein that sticks out of cells. And they were able to find out that what these fibrils are made of is this little sequence right here. So there are molecular scissors that come in and actually cut at these 2 points here and release the A beta. You have A beta floating around in your fluid that bathes the brain right now, as well as the spinal cord. So this was fairly helpful in really setting the course of the disease and thinking about what's the role of beta amyloid, and they did the same thing in terms of tau. So I illustrate this because, if you notice this little cartoon here is the same one that I had in the previous slide. This gene that's on here is actually on chromosome 21, which Down syndrome individuals have an extra copy of. So they're getting the gene that produces this beta amyloid that produces the plaque. So from birth, they are actually exposed to this protein. And they do develop some of these protein buildup by the age of 30. And the sad part of the story is the fact that these individuals normally didn't live past 20 or 30 years of age. Medicine has gotten so much better at treating them that now they're living into their 5th and 6th decade. Well, now they're facing Alzheimer's disease, and a large number of them develop dementia. So for that reason, the Down syndrome community is actually trying to rally their constituents to realize we're getting past this, but did you know that Alzheimer's is a problem for these individuals? Plus, they provide an excellent opportunity to understand why does that extra copy of that gene cause Alzheimer's disease. So helping us. And they also realize that maybe some of the same treatments that we will be giving individuals for Alzheimer's disease could help the Down syndrome individuals. So this has been kind of a labor of love working with this. I put together a workshop to help bring this together. We put $1.2 million into a joint initiative with the Global Down Syndrome Foundation. So for that reason, we're trying to work together. Another way we're trying to convene people. This is just to point out, it's a complicated graphic, but This cartoon showing this A beta is that in the protein structure here, is that where the red letters are, that are individual changes in amino acids, represent a mutation that causes Alzheimer's disease. If you have one of those mutations, you get Alzheimer's disease. We're lucky that it's a very low majority of people that have these. But they've been very helpful in trying to understand this. The reason I put this up here is because this little A beta sequence up here is either made, there's more beta by where it's being processed, or it helps fold the protein into this abnormal shape and cause the clumping in some way. This has been helpful. We've learned a lot over the last 20 years about Alzheimer's disease. We're not there with the treatment yet, but these things have been extremely important in understanding mechanisms. So I also underline this because we're still talking about certain genetics, that what we found is there are genes that enhance your risk. They're finding more of those all the time. They actually probably have relatively low effect, but maybe in combination is what puts you at greater risk. There's only one or two genes that have a major effect. We call one of them is ApoE4. But some of these other genes may be in combination with other genes that affect it or interacting with the environment. In other words, we talk about blood pressure. We talk about cholesterol. Those sorts of things may affect this gene profile. But also, we're finding genes that may put you lower your risk for the disease. There's the other side of that. We want to learn that aspect of it. Just like we're learning from Down syndrome individuals, we want to identify that people who are at less risk. Maybe they can teach us something about treatments. And so we're going to move towards clinical trials, but I have to put this up just to set the stage to think about where we are. And people always ask, why don't we have treatments now? Why does it take such a long time? Well, you can see from here, it's almost about 13 years to start from the very initial discoveries that happen often in academic labs that are here. Then they get transitioned over into pharmaceutical companies. And then at some point, they have to test it in humans. So this stage is usually laboratories, maybe animals, getting some information. And then phase 1 and phase 2 trials are really trying to understand the safety of a new medication. In other words, they want to find out whether they're going to dose you at a certain amount. Does it produce more harm than good? So that's the first stage, is safety. The second part of that is, does it do what they want it to do? Does it change memory? Does it change, the progression of the disease? What is its efficacy? That's usually in stage 2. And phase 3 is finally putting in a large enough people to say that you have confidence that it's safe and it's effective. And it has to be done in 2 separate trials in order for the FDA to approve it. So right now in phase 1 and phase 2, we have a number of drugs in there. The problem is that there is a number of places where this whole process failed. One is in the pre-discovery phase. Often we see a 90% failure between the transition to pharmaceutical companies and a 90% failure at phase 2B trials. And this needs to be changed to make sure that there's a much greater efficiency about the number of drugs that are going to reach the end of the pipeline. So this is just to explain that there's a wide profile of drugs being looked at this point. We will talk about amyloid or A beta, we'll talk about tau, which has been a focus because of what we see pathology-wise, but people are still looking at other mechanisms. And this is where we are with phase 3 trials. And the 2 that I can point out here are some of the new trials that are going on in terms of we're looking at tau, Rx, and then also the solanezumab. And solanezumab was one of the clinical trials that recently reported out. So in the last year, we've actually had 3 major clinical trials reported. One was IVIG, or gamma-guard intravenous immunoglobulin. This is giving an antibody taken from healthy individuals and then put into mild to moderate Alzheimer's individuals and seeing whether it changed the course of the disease. It had no successful outcome. Even when they drilled down into the data, they saw no promise in IVIG. You may have noticed, actually, there is another company that's working with IVIG, but they're also mixing it with another substance to see if that would actually change the efficacy of it. What I want to point out about this one is that every clinical trial has to have its top-line outcome. That's usually looking at cognitive impairment, your thinking ability, and for that reason, your functional, your dementia, your daily living activities. Those are the two primary outcomes. They failed both of those. But in a pre-specified analysis, they said, we're going to take both of those clinical trials and put them together. We can pool a lot more data. We can better statistics out of this. We want to see if there's a difference. And the first time, they saw actually a 34% reduction in the cognitive decline. Now, what's interesting about that is that this was always done in mild to moderate individuals. The most benefit was actually in those that had the mildest form of Alzheimer's disease. So it started thinking, which is also going on at the same time in the other parts of the Alzheimer's community, is the question, are we going early enough with these particular drugs? These are what we call anti-amyloid. They're going out to the A-beta. And these are actually using an antibody, something that we all have floating around in our blood, that help go after foreign materials to clear it. And this is a passive immunization, meaning that they're giving you the antibody as opposed to you making it. Just like why you get a tetanus shot, so that you can produce your own antibody against tetanus bacteria. So for that reason, it's the idea that our immune system can work to our advantage. These have been designed to particularly take away beta amyloid. So the advantage about this study is the fact that even though it did not work, what you see here is Eli Lilly decided to go on and do their Expedition 3 trial. It's an extension of this. But they learned from the first part, and they're only going to enroll people that are the mildest form. And they also want to know, do they have amyloid in the brain? Does that make sense? Because if it's anti-amyloid therapy and there was no amyloid in the brain, then that's a problem. And in fact, we do see from our imaging studies that there are people that have amyloid in the brain that show no symptoms. And there are people that show symptoms but have no amyloid. It's about 20 to 25% that we see, and maybe the end of the spectrum. So there's a lot of people still treat with amyloid in the brain. But that's one of the things that they're trying to do. I also want to point out that it's particularly important that we make sure that these drug companies stay engaged. Now, yes, they are for profit, but they are the ones who are going to be delivering these drugs to the community. They can sustain some of the large costs in the clinical trials. Now, they have actually had problems getting drugs in the central nervous system, which has been a problem for them. But I think Eli Lilly is in for the haul on this, and we do appreciate that. So one of the questions is, when does the disease begin? And I mentioned in terms of Down syndrome individuals, they're born with an extra copy of chromosome 21. So what we'd like to know, as in the question about the solanezumab, the drug that I was just talking about, do we want to go earlier? So in 2011, they came out with some new criteria for the diagnosis of Alzheimer's disease. And it was trying to talk about, you don't have Alzheimer's disease on the day that you're diagnosed. The question is, when did you actually begin the disease? We understand this from a lot of other disease perspectives, that there's actually a continuum. In other words, there's a normal state and then there's Alzheimer's disease. The question, what comes in before? In 1984, the diagnosis for Alzheimer's disease was seeing plaques and tangles in the brain. We want to move to a point where we can actually understand this at a much earlier stage. So they talk about the 3 stages here in terms of dementia, mild cognitive impairment, and this preclinical phase, or the silent phase. I like to think about it really from the standpoint of cardiovascular disease. Why do you get your cholesterol checked? Because high levels of cholesterol can possibly lead to plaques building up in your veins, and that can lead to cardiovascular disease. So for that reason, do you know that you have plaques building up in your veins? Probably not. It's unfortunate that most of the time people realize that when they either have some complication like the heart attack, etc. So for that reason, understanding that puts you either at risk or development of the disease. That's really what we're thinking about in terms of the silent preclinical stage. But if you don't have any symptoms for the disease, how do you know that marker is actually telling you that you're going to get the disease? And that's what the cardiovascular field did for many, many years, is trying to work on why is cholesterol a valuable marker, a biological marker for the disease. So this is really a very active area in the field because it will do two things for us. It will tell us, first of all, whether that's a marker for the disease, and will also tell us whether a drug is working. So if you're in a silent phase and you give a drug to somebody, how do you know that in fact that is changing the course of the disease? You won't know that until either it's clinically present or that you can see it's changing. So this is what I wanted to talk about in terms of cholesterol. There was the war on it, trying to figure out what was good and bad. And the fact that it was able to reduce maybe morbidity by 25% through these sorts of treatments. That's why people often get statins these days, is to lower their cholesterol levels. Well, hopefully someday we maybe have a drug that would lower the beta amyloid levels or the tau, or what other agent that we think is important in those early phases of the disease. So, and I think one of the telltale things here is delaying dementia just 5 years to decrease Medicare costs by 50%. So that was that $203 billion I talked about. That's a significant saving. Also, if you can delay it long enough, somebody may actually have a normal, happy lifespan. They may die from other causes. So for that reason, delaying it can be just as important as stopping it or curing it. So we have a number of biomarkers here. They're particularly looking at, there's a number of imaging techniques, and one of them I want to go through, well, we'll stop here for a minute. One is actually looking at those proteins. I talked about beta amyloid, and I talked about tau that are components of the two hallmarks of disease. Those are actually, you can measure those in your cerebral spinal fluid. That's the fluid that bathes your brain and your spinal cord. So they can do a lumbar puncture to go in and actually look at those proteins. And they actually are a very good marker for understanding the progression of the disease itself. As you can see, it's, somewhat invasive. People do not necessarily like to think about lumbar punctures, but apparently in Europe they do it much more commonly than they do here. But it can tell you a lot about the progression of what's happening to amyloid, what's happening to tau. But I also want to focus on something that's been in the news a lot, is amyloid imaging. This is the ability to give someone a drug that's radioactive, give it into a vein, it goes throughout the body, and what it does is actually bind to where amyloid is forming. That's in the brain. And what you can see here is images of the brain. And as someone is developing the disease, this lights up. And for that reason, we can actually follow the person in terms of their progression. Over here is actually the data that showed that indeed, when you look at the tissue from somebody that actually died, that had one of these scans, they have these plaques in the brain. They can actually tell that it's building up. This is normal. This is someone with Alzheimer's disease. So now we have a non-invasive technique to look at what's happening with this particular pathology. And for that reason, we formed the Amyloid Imaging Task Force to look at appropriate use criteria for this. And it's important because we feel there is specific people that this test would be of value, but it's not for everybody. This test costs between $3,000 and $7,000, depending on where you're at. It's very expensive. Center for Medicaid and Medicare Services has not decided yet to pay for this test. In fact, they want more data. They have something right now coverage with evidence development, meaning that we're going to have to design clinical trials to prove that it does change clinical management or patient outcomes. That's one of the things that we're trying to do. But this was to specify that it could help in the diagnostic accuracy to those people that actually may look like they have Alzheimer's disease, but it's difficult. Somebody who has cognitive impairment by a objective criteria of some sort, looking like they're progressing in some way. But we want to find out whether they have actually amyloid in the brain or not. Most cases, this is a tool. It does not diagnose. It's not diagnostic for Alzheimer's disease. What it does is rule out the role that you have amyloid in the brain and gives greater certainty in terms of that diagnosis. So right on the heels of the imaging for the beta amyloid in the brain is tau imaging. This is really the cutting edge of thinking about this, and we've just seen a number of, at the clinical trials for Alzheimer's disease, presentations about how tau is being used right now and very close to being tested. And what you can see again is that it's going to tell you about the development of tau. And I think that's really critical because this was something that was recently produced by Keith Johnson, who is a imager at Mass General Hospital. And here is something looking at amyloid, and here is something that's looking at tau. You can see that there's differences in terms of the overlap of these patterns. The important part of this is that these tools together could give us a better diagnosis for Alzheimer's disease. More importantly, it may tell us how it progresses through the brain and where these two things overlap. It may be critical where the two pathologies come together to actually produce the disease. Something that we need to know more about is how tau and beta amyloid perform in the brain in normal aging. And it's seen a lot in the elderly brain. So for that reason, we're trying to figure out when does it convert to Alzheimer's disease. So one of the things that we've orchestrated here is worldwide ANIME, which stands for Alzheimer's Disease Neuroimaging Initiative. And it's a way to standardize how these images are taken across the globe because we think that's important. So as we look at studies, we can compare apples to apples instead of apples to oranges. In other words, who's running in terms of the scanners, how is it being done, how is that standardized? Who's reading the scans, how is that standardized? So this is something that we've been doing to make sure that we're on track for comparing these, and we often have many meetings that are run that somebody's up in the middle of the night to make these phone calls to discuss data. One of the things that we're trying to do is also make data available, freely available to all researchers. I just talked about ADNI, and for that reason they are making their data available. We've just sequenced all 818 individuals from ADNI in terms of their whole genome sequencing. It's a very enormous amount of data, but it can be mined for understanding about how some of these imaging studies, how some of the neuropsych tests that are being done correlate with the genes that they're seeing in these individuals. This will actually not be just mined probably by the Alzheimer's field, but it'll probably be mined by cardiovascular field, by the other fields, because this is one of the largest groups to be having whole genome sequencing. So this is something Alzheimer's has put some $5 million into because we feel it's important to share this data, and we'll continue to do that to make sure that researchers have some of the latest information, as well as that they want to share their own data. They have a platform for doing it. So this is a hypothetical curve just kind of talking about biomarkers and when do they show up. So this is time, and we can just take something like beta amyloid. So this imaging that I just talked about is the red line. But sometimes it's fairly low and then increases. And here are the disease states: normal, mild cognitive impairment, and dementia. So the question is that are any of these biomarkers preceding the development of the disease? And this was something, again, that was put out about 5 years ago. And I bring that up because I mentioned the Eli Lilly trial. If you recall, the people that were most affected by that drug were the mildest. So that's another piece of thinking about, for those drugs, should we be thinking about going earlier? Will those be the most effective? We're just using them at the wrong time. So DIAN stands for Dominantly Impaired Alzheimer's Network. These are those individuals. I showed you a graph where you change the single letter and it comes from various countries. If you have one of those changes, you get Alzheimer's disease. It's a familial mutation. Again, it's fairly rare. But for that reason, there's a study at Washington University in St. Louis looking at these individuals. And they come from all around the country, and now they're actually coming worldwide to this study for 2 reasons. One, we know they're going to get the disease, and we know about when they're going to get the disease. They often get the disease very close to the same age as their parent. So those are 2 pieces that are very powerful in a clinical study to be able to say who and when they're going to get the disease. So they did these studies of looking at logical memory and, whoops, we'll go back here for a minute. looking at logical memory. And the red line are those people that have mutations, and these are not. This is stable here. But you can see their memory changes almost 10, 15 years before the onset of the current clinical symptoms. If you look at beta amyloid, we're almost 20 years before the clinical onset of the disease. It's building up in the brain. That's important because if beta amyloid is helpful in thinking about initiation of disease, that's a 20-year window in there. So for that reason, we think we have a window of opportunity to possibly treat. So these pieces have now been the verification of that hypothetical curves, and we're learning more about that. We'll do the same thing with tau as we get more imaging online. What is the timing of these biomarkers? When do they occur? And for that reason, it may be very important that at some point, maybe beta amyloid doesn't matter anymore. It's done its job, it's moved on, and now there's another process that's involved. For that reason, you may not want to treat somebody with beta amyloid at that point. It may be totally inappropriate. So I titled this, Back to the Future, because the question really is, I mentioned that we had, a goose steer's brain. We saw the plaques entangled. That was the disease itself. The question is, how far back do we have to go to actually find an answer in terms of the disease itself? So I call it back to the future because we're going back to the beginning of the disease, but the future is, can we treat them? Can we think about prevention? So this is just a slide to look at really kind of where we are right now, thinking about tertiary prevention here. We're in the disease state itself, so it can delay onset or progression of the disease itself. That's been very difficult for us to see any movement in. The reason why, there's probably a lot of things going wrong in the brain at that time, particularly things like cell loss, disconnections between cells. That's difficult to repair. It may mean that we're going to have to move towards combination therapies in order to have this be effective. And we've had discussions with the FDA of how we might move forward in terms of clinical trials to think about doing that. But as we move, further to the left here, is that we're thinking about secondary prevention. In other words, this is that silent phase in here where we're seeing the biomarkers change. That may be the way that we can identify people who's at the greatest risk for the disease itself. Then hopefully someday we can think about primary prevention. There may be things, even in terms of lifestyle, that we may be able to change that would lower the chance of even developing any of those first biochemical markers. So I like to look at it this way, is that if you start to treat here in terms of this is the kind of cognitive decline over time in someone, that if you treat here, you may move the curve in some way, but they're still going to develop dementia. It just may be delayed. But if you go somewhat earlier in this stage here, what you're going to find is maybe you're changing this curve to the point where they don't develop the dementia, but they still may have mild cognitive impairment. And if we go even earlier, the question is that can we get this flat before they're behaviorally effective in some way or clinically effective? So that's really what we're thinking about at this time, and that's here now. There are now 4 prevention trials that are either underway or being designed at this time. And the idea is to take some of those drugs that took a lot of time and effort and dollars, and can we take them and move them earlier in the disease state? Such as I talked about solanezumab from Eli Lilly, crenezumab that's coming from Roche. Those are drugs that are out there that will actually be used to see if we can change the curve. And the A4 trial, anti-amyloid asymptomatic Alzheimer's disease, is something that's being done between Mass General Hospital, Brigham and Women's, and the Alzheimer's group that's at University of California, San Diego. And A4 is unique in the fact that what it's going to do is look at individuals age 65 or older that have amyloid in their brain but are cognitively normal. In other words, by our standard test. And the idea is to treat them because we think they're at risk and treat them before those signs develop and see if that mediates or slows down the progression of the disease. The API is actually looking at other mutations. There's a large family in Colombia that's being studied. It's almost a family of like 5,000 individuals, and like half of them develop the disease itself because they have one of these rare mutations that causes Alzheimer's disease. So for that reason, they've been studying this family for some time, and now they're going down there and seeing if they can actually treat them with an anti-amyloid therapy. This is being done out of Banner Healthcare out of Arizona. And they're actually now recruiting for people that have one of those genes, ApoE and ApoE4, that puts you at great risk. And they're now trying to develop a trial which they're going to enroll in people with ApoE status. Tomorrow is one that's looking at ApoE as well, and they're also using another gene called TOM40 that shows when you're risk for developing the disease. So what they're doing is stratifying, in other words, gathering those people that are most vulnerable to the disease itself. It helps in terms of that population, and there's a statistical analysis to make sure that you're going to understand whether your treatment is working or not. And I mentioned about DIAN because it's one that we put $4.2 million into. It was our first large grant that we put in. And the reason why is because we think it will be a watershed of understanding whether these anti-amyloid therapies really do change the course of the disease. You know, we can hope that, in fact, that it will stop the progression or never develop the disease, but we can also hope that maybe it could slow down the disease. That would be a win as well. So what we're going to do is look at, I call this kind of a plug and play. It's brand new in terms of adaptive design. We learned this in the cancer field. And what they're going to do is put at least 3 different drugs into this population. So the fact is that what happens is that there are 3 arms to it, in which they're all getting treatment, and there's only 1 placebo. So for that reason, most people are actually getting treated in this trial. And what they're going to do is first look to see if any of those drugs actually change the biomarkers. In other words, the amyloid, the lumbar puncture, looking at tau and A-beta. And they're going to see whether those drugs start changing how those progress over time. If we're lucky and they actually do work, then they'll move on to the next phase and do the standard analysis of looking at cognitive and functional outcomes. If for some reason they do not move those biomarkers, they'll be taken out of the study and a new one will be plugged in. That's why I call it plug and play. They don't have to start that trial over again. They actually have a pharma consortium that's working with them and thinking about what's the next one to plug in. So they're hoping that this will actually accelerate the clinical trial process of looking for some effective drugs. But also important is this, that those studies will help us validate, is it a proper surrogate that one of these biomarkers is saying that, yes, you're at risk for the disease and you're developing the disease. So it's a 2-part study, and that's one of the reasons why we put this $4.2 million. And one of the reasons we also did it is that there are 10 pharmaceutical companies working together on this project. There are certain things that they cannot do in terms of funding somebody else's trial. Our infrastructure helps making those people come together and play in the same sandbox to do this sort of analysis. So that's one of the reasons why we did this, and we actually did this almost 11 to 12 months sooner than they got the NIH funding. So we wanted to move forward. So this is just to indicate too that there's always a question of are using the same things in the same different trials. So between the A4 trial, the API study tomorrow, and DIAN, we actually have been co-leading with Fidelity Biosciences, and we actually get together with all heads of these 4 different studies to talk about what cognitive assessment are you doing, what type of biomarkers are you using, how are you assessing this. So it's a way to make sure that when these studies finish, that they have an idea that they're comparing studies together. They may even be able to pool some of the data together. So that's why we're leading CAP here, this coalition for Alzheimer's prevention. And one of the things I want to bring to your attention is something that we do about trial match. This is a free service that gives you the opportunity to enroll in a clinical trial. Free service you can either do online or through our helpline. And with that, they can find a clinical trial that's in your neighborhood. And it's an idea that you'll probably enroll and stay in that if it's in your neighborhood. We know that when we list patients to clinical trials is getting full enrollment. There have been a number of cancer trials that never went forward because they could never meet their enrollment. So it's participation by volunteers that's critical for clinical trials. So I'm going to finish up by saying, what do you do now? Well, hopefully you got the idea of support research. That's really critical. It's the only way that we're going to stop people from having Alzheimer's disease. Second part is talk to your legislator. They are the ones that can move the needle in terms of the dollars that are going to be needed for that research. In other words, they fund or they set the appropriations for National Institutes of Health. You can actually make a difference. If you're voting for that legislator, they often will listen to you. So tell them about your concern for this. I gave you some numbers earlier on, and you can download all of that from our website. But if you ever have an opportunity to talk to the legislator, take some of those numbers with you. Join a clinical trial. I just talked to you about trial match, an easy way to do that. Another way is stay healthy. Know your numbers. Think about your cholesterol levels. Think about your blood pressure. Think about your glucose levels. Those seem to be risk factors that we're trying to understand more about. Clearly, staying healthy is a great way to do that. And stay engaged, both physically, mentally, and socially. We don't know whether those will change the course of the disease, but what it can do is make a healthier brain. For that reason, the question is, will that either slow the progression of the disease or in some way put you in a better position to deal with the disease itself? So for that reason, these are things that you can do now. And with that, I'll finish and say thank you, and I appreciate you coming.